Hello, everyone. I'd like to welcome you to our genetic research education series with a focus on TARDBP. This webinar today is brought to you by Genetic ALS and FTD and the Legacy. We are a patient-led organization proud to serve our community. We strive to serve the needs of the community through education and support services like today's webinar. We also serve to represent the interests of our community in dialogue with the academic fields that study us, our government, and the industry developing drugs for us. My name is Dr. Nadia Sethi. I joined in the Legacy several years ago, and I am now a very active member. I am very excited to bring our content today to you on TARDBP. And with that, I'm going to go ahead and also do a few acknowledgements. We would really like to thank our speakers today for being here. It takes time and effort to do this, and for them to give up their time to be here today. We are very, very grateful. We would also like to thank Biogen for sponsoring this series and for supporting And the Legacy. And we'd also like to thank everyone who funds, conceives of, and carries out and participates in the studies that we're going to be talking about today. You are the people who are helping to move the research forward for all of us. So thank you so much for all of that. On today's agenda, we have Elizabeth Harrington, who is a genetic counselor at Columbia University. We have Bonnie McKay Harris, who carries a TARDBP variant. We have Sean McCann, who is living with TARDBP ALS and will be sharing some of his research experiences and a little bit about himself. And we have Dr. Neil Shneider from Columbia Neurology, who will be talking about research for TARDBP. And then we will close everything out with a Q&A. Please feel free to share questions in chat. And also, we will be posting this recording on our website. You can find our previous recordings on SOD1, C9orf72, and progranulin at our website, andthelegacy.org. And with that, I'm going to stop screen sharing. And I would like to welcome Elizabeth Harrington, first of all. Elizabeth Harrington received her master's in human genetics and genetic counseling from the Stanford School of Medicine. She is a neurogenetics clinical and research genetic counselor with expertise in motor neuron disease. She joined the Department of Neurology faculty in 2019 and is a lecturer in genetic counseling. She directs the ALS Families Project research study, a pre-symptomatic natural history study designed to understand the genetic underpinnings of genetic forms of ALS and the impact on affected families. She is also the preclinical Silence ALS program genetic counselor and coordinator, and we're going to be hearing about that a little bit later today. So Ellie, I'm going to have you take the screen, and we're very excited to hear from you. Thanks for being here today.
Great. Thank you so much. Thanks everyone for being here today. For my part of our conversation today, I'm going to really start with a basics overview of genetics, ALS genetics, and dive into TARDBP genetics. And then we'll talk a little bit about the considerations for genetic testing, and I'll end with a little brief introduction to some of the research we have. But hopefully all of what I talk about today will really set up Dr. Shneider to go more into the scientific details of some of the programs we have ongoing at Columbia. So give me a moment while I share my screen. Okay, does everyone see that okay? All right, great. Okay, so getting us started on very basic genetics, most people, this relates back to high school biology. So obviously humans are a very complex being. We're made up of trillions of cells, and each of our cells contain our complete genetic code, or our genome. When we zoom into that, our genome is held on what we call chromosomes. And when we zoom into our chromosomes, we can see the specific spelling of our DNA code. So that's made up of DNA nucleotides, or A's, T's, G's, and C's. And portions of your DNA sequence spell out specific genes. Each of our genes have specific roles and functions in our bodies and our cells. So this DNA is transcribed into messenger RNA, which is then translated into those proteins that have those specific functions in our bodies. Most mutations in genes that cause disease, you can think of like spelling mistakes, so a change in a single letter of that DNA sequence. And the result of that can be a mistranslated messenger RNA and resulting protein. So either dysfunctional protein in many different ways in the cell. Continuing on with that basic genetics, we share our genetics within our family. So everyone probably remembers the doomed Punnett square you did. But our genes, we have 2 copies of all of our genes held on our chromosomes. So when we're inheriting genes in our family, we have you pictured here on the left of this figure and your partner. You pass down one of each of your 2 gene copies, and your partner does the same. Most of the ALS-associated genes are dominant in inheritance. So in this example, your partner does the same as previous, passes down one of their 2 normal gene copies. But for you with a mutation, there's a 50% chance you pass down the mutation and an equal 50% chance you pass down your normal working gene copy. So autosomal referring to males and females being equally affected, dominant referring to the 50% chance that that's inherited from a parent. You only need one of your 2 gene copies for one to be at risk for developing disease. As we get into ALS genetics, if you do any reading or Googling online, most of the terms that we see are sporadic and familial ALS. Frankly, these aren't very scientific terms. They really refer to the presence or an absence of a family history of ALS. So sporadic, meaning there's no family history of disease. And when we do genetic testing on somebody with no family history, it's less likely that we identify a single genetic cause. The chance of that isn't 0, though. Still about 5 to 10% of people with no family history still have an underlying gene mutation. For those that do have a family history of ALS, familial, they're more likely to have a genetic cause of disease. So about 80% of the time when we do genetic testing on somebody with a family history, we're able to identify an underlying gene mutation. There's been about 30 genes that have been associated with ALS. So back to our figure, our genes code for that specific protein and function in our body. So many of these genes are responsible for protein transport, RNA metabolism, mitochondrial functioning, DNA repair, which all help in your healthy motor neuron functioning. So when we have a mutation in one of those genes, those typical functionings aren't working properly, and that leads to motor neuron degeneration. So TARDBP makes up about 5% of the genetic causes of ALS. So the most common gene that most people have heard of, which I think has been covered in these webinar series, is C9orf72, followed by SOD1. And here at the bottom, I have a figure of this TARDBP gene. I like to describe it kind of like a long sentence that spells out that recipe or function for your cell. And you can have a mutation at any letter in that sentence or any location within that gene. So these different letter and number sequences down here are just the names of different mutations within the gene. And over 50 mutations have been identified in TARDP that have been associated with ALS. TDP43, or TAR DNA binding protein, is the protein that's produced from this gene. So this really plays a crucial role in neuronal health, stress response, and RNA processing. When a mutation occurs in this TARDBP gene, it creates an abnormal gain of function mechanism. That means that instead of creating that normal functioning TDP43 protein, it creates these abnormal protein aggregates, which really disrupt the normal motor neuron functioning and contribute to motor neuron disease. In addition to its association with ALS, we also know that TARDBP DBP can cause frontotemporal dementia, or FTD, which is a much more behavioral dementia. So people see changes, increased impulsivity and inappropriate behavior, cognitive decline, including impaired judgment and planning, language difficulties, among other things. Continuing on in one's risk for developing disease and risk associated with a specific variant, we always talk about penetrance. So penetrance refers to if somebody has a gene mutation, what's the actual likelihood they might develop disease symptoms? So there are some gene mutations, like the gene that causes Huntington's disease, that's 100% penetrant, meaning 100% of people with that mutation develop disease symptoms in their lifetime. Many of the mutations that cause ALS, including TARDBP, have reduced or variable penetrance, meaning you can have the mutation, but perhaps there's an 80% lifetime chance you'd develop disease symptoms and 20% chance that you wouldn't develop disease symptoms. So this figure here on the right is one that can be used in genetics. We have a baseline population risk. When somebody reaches this red line in the jar, that's the point or threshold in which they develop disease symptoms. For ALS, we know there's likely a combination of genetic factors and environmental factors that contribute to the onset of disease. For people with mutations, they're kind of born with this big genetic factor. So a big genetic factor that has reduced penetrance is one that's shown in the example here on the right, where they're not born already at that threshold where there would have been 100% likelihood to develop disease symptoms. As we get into genetic testing for ALS, I'll briefly touch on diagnostic genetic testing, but also a lot of what we're focusing on is predictive genetic testing for asymptomatic individuals. For diagnostic genetic testing, this means genetic testing for people with disease, looking to see, is there a genetic cause for your symptoms? There are now published guidelines for practitioners to recommend genetic testing to everybody with ALS. And this includes genetic testing for C9, SOD1, FUS, and TARDBP, so kind of the more common genes. Most of these genes are all tested for at once on one order. So if you are somebody living with ALS, genetic testing is recommended, and you should speak to your provider or genetic counselor to discuss the pros and cons of testing for you and your position. I want to dive more into the considerations for genetic testing in individuals who are asymptomatic. They might be at risk for developing disease based on their family history. So perhaps there's somebody whose parent has TARDBP ALS, and you yourself are considering genetic testing. There's a couple of different things that I always urge people to think about as they're going forward with genetic testing themselves. So these are things that we talk about in a pre-test genetic counseling appointment. People have a lot of different reasons why they choose to move forward with genetic testing. What's important is that those are kind of well defined for you. What are you going to do with this information? So some people say, I would change my career. I want to plan differently for my future financial planning. They have better self-advocacy. They are better aware for potential medical concerns down the line. They want to be screened earlier, which can lead to an earlier diagnosis. People consider their family relationships. They might want to change family planning, use in vitro fertilization and embryo screening with genetic information in mind. They might want to clarify risks for their own children. So this is something that is certainly talked about and considered when choosing to undergo predictive testing. A lot of people also consider the community, participation in research, contribution to observational research, some of which we'll talk about today. There are certainly, on the other side of things, some concerns that people have with testing. So remaining anxieties and uncertainty can exist with a genetic result. Of course, if you test negative, those anxieties and uncertainties are minimized. But if you test positive, there's remaining questions. Will you get disease? What will that look like, ALS or FTD? When would that be? How might it progress? Those are questions we don't have answers to right now. And people might have increased anxiety after they know for sure they're at that increased risk. We also talk about family and relationships. So who are you planning to tell this result to? How might it impact them? How might you communicate that information to your children, to your siblings? People have, there might be feelings of parental guilt of passing mutations on to their children, or survivor's guilt if one sibling tests negative and one sibling tests positive. The last thing we always talk about is genetic discrimination risks. So we are all covered by a law called GINA, Genetic Information Non-Discrimination Act. This law protects all of us against discrimination from our health insurance companies and our employers if we work at a company more than 15 people. It, however, does not protect against life insurance, long-term care insurance, and disability insurance. So it's important, if you're considering predictive genetic testing yourself, that you make sure those insurances are all in place and you feel comfortable with those before you undergo predictive testing yourself. So these are all considerations that are going to be very different to each individual. I talk to large families, and each of these are different conversations among siblings, all testing for the same mutation, and depends on where you are in your life stage. So we recommend that you discuss these questions, these topics with a genetic counselor who can walk you through the different options and opportunities for genetic testing and potential research involvement. Talking about research involvement, I'll briefly mention the ALS Families Project and Prevent ALS, which is observational research participation for asymptomatic individuals at risk for developing ALS in the future. So the ALS Families Project is our historical study. We've started in 2018, led by Dr. Shneider and Dr. Harms at Columbia. And we are kind of merging that into Prevent ALS, which will be a multi-site study, I think 34 sites around the US. So very similar studies, very similar goals. Prevent ALS is kind of this larger study that is ramping up at different sites throughout the US over the next couple of months. But the goal of these studies is to characterize the pre-symptomatic disease state, gather natural history information, including biofluid collection for individuals who are genetically at risk. So we do that by performing annual research evaluations. People come into Columbia once a year. We collect blood samples, urine samples, skin biopsy, perform neurological exam, cognitive testing. So series of clinical evaluation and biospecimen collection over time to really reach these goals of learning more about the genetic forms of disease to be better able to create targeted therapeutics. Through this study, you also have the option to undergo predictive genetic testing with return of results. So you'd meet remotely with a genetic counselor, myself and two others, who will bring you through that genetic testing process. So that's an option. If you are at risk, you still have the option to participate in the main study without learning your genetic results. So you can remain a non-disclosure participant, blinded to your personal genetic status. So from this, we've really, from the Families Project at least, we've really have been able to identify large families with more rare gene mutations. So Dr. Shneider will really dive into the Silence ALS program, which will talk about our goals in creating ASO therapeutics to target these more rare genetic forms of disease. So just a very brief introduction to ASO therapies. We've kind of seen this figure, our DNA to mRNA to our protein. When there's a mutated DNA strand, we have a disease-causing protein that occurs. So an ASO is a type of drug that's designed to target your messenger RNA here and basically latch onto it like a tag and put a stop signal on it, so it can't create that disease-causing protein. So these are successful therapeutics that have been FDA approved for other genetic diseases, including most recently SOD1. The drug Qalsody was FDA approved. And there's others in clinical trial, notably the fusion trial for the FUS ALS. So with that, I'll kind of end. I know Dr. Shneider is going to dive more into the Silence ALS program, which is developing those ASOs for TARDBP specifically and other more rare genetic forms of ALS. So I'll let him take the stage. I know we have a few other presentations in between now and then, and I'm happy to take questions at the end.
Nadia, you're muted.
Live entertainment. Yes, next up, I'd like to introduce my friend Bonnie McKay Harris, who carries a variant of TARDBP and has participated in multiple research studies and will share her experiences with those studies and with living with this variant. Thanks so much for being here, Bonnie.
Thank you. Thank you for having me, and hello, everyone. Like Nadia said, my name is Bonnie, and I'm going to walk you through my experiences with longitudinal studies, these ALS studies. So my story begins in 2017, and my dad was diagnosed with ALS after almost a year of symptoms and numerous doctor's appointments. My grandmother, my dad's mom, had dementia, and her brother had ALS. So we knew that this disease plagued our family, but we had no idea that dementia was even tied to this disease. My dad saw Dr. Bedlack at Duke, and he discussed with us the relationship between dementia and ALS and recommended the genetic testing. The results soon came back that my dad had a mutation in the TARDBP gene. In my mind, we now had something tangible to work with, something to go after. So I read every research paper that I could get my hands on that said TARDBP or TDB43. I emailed all the doctors that wrote these studies, and they were located all over the world. I was asking them, please fix my dad, somebody do something. And we didn't get that fix that we were looking for, though. And 14 months later, my dad passed away. What I did get, though, was connected into observational research studies. So the first study that I joined was at UPenn called All FTD. And this study is where I did my genetic testing. I went in for my first appointment, and they took some background information, asked me a ton of questions, and then took some blood, and we knew which gene that we were looking at. And then the next appointment, I found out my genetic status. So then at the following appointment, I started going through MRI scans, blood work, extensive cognitive testing. It's a 2-day adventure, and you bring along a study partner. So in my case, my husband comes with me, and they ask him a series of questions about my mental health as well. So I have to be very kind to him leading up to this appointment. This study does pay for travel, and it's once a year. So I get my hotel paid for. I get, we drive there. So our gas money is reimbursed, and then any food that we eat while we're there, but they also can reimburse for your airline. So then the next study that I joined was the DIAL study at Mass General. And this study is now transitioning into the collaborative study, All ALS, with an arm of that called Prevent ALS. So this study does in-depth strength testing, and that's on a machine called the Atlas machine. You go through some cognitive testing, but it's not as in-depth as at UPenn. You have blood work, and then I have an optional lumbar puncture, And the lumbar is done annually, but the visits are every 6 months. And if anybody's nervous about a lumbar puncture, you don't feel it. So it is not this big scary thing like I initially thought the first time I had one done. But they have allowed me to spread out my visits a little bit further apart because I don't like to go in the winter. And so they kind of let me schedule things and give me some flexibility. There is no travel reimbursement for this study. And so when I go to Boston, I fly out of Raleigh, North Carolina. I fly into Boston in the morning of the appointment, and the appointment lasts about 4 hours, and then I fly back out that evening. I was also able to join Silence ALS at Columbia, and a sharing agreement was set up with Mass General so that at my dials appointment, they were able to collect what was needed and send it over to Columbia. So I'm telling you all this to let you know that how incredibly accommodating everyone is. I just can't emphasize that part enough. Lastly, there's the ARC study that was sponsored and managed by ALS TDI. This study is done from the comfort of your own home. ALS TDI will have a phlebotomist come out to your home. They take your blood. You answer a series of questions on your well-being. You do some voice recordings, and you wear a wrist monitor for a week each month. And this tracks your activity. This one is super simple. If you don't have a ton of free time, it's an easy one to participate in, and there's no out-of-pocket expenses. So all of this is to say that joining these studies has been a really, really positive experience for me. The investigators and all the staff at the studies have been so pleasant to work with. I even have a genetic counselor that calls me and checks on me on occasion and just to chat, and we just catch up. So it's a very comfortable experience. I feel like by joining these studies that I'm not sitting there waiting on ALS to come get me. I am going after ALS. And I have an amazing team of doctors and researchers that are working so incredibly hard to find a treatment or a cure for all genetic carriers. It only takes maybe a day or two max of my time for a study visit. And so it's not that much time to give to these doctors that are fighting so hard. So if none of these are close to you, these studies are close to you, like there is the pre-falls in Miami. And then, like Elizabeth said, the All ALS is expanding to 34 study sites. So that hasn't happened yet, but that's their goal. So this is going to give a lot easier access to participants. Being involved in these studies, it really gives me some hope. And I know that there's some really smart people out there and they're on our team. So that's so refreshing. They're working so tirelessly to find biomarkers, treatments, and a cure. And we need that. So please, if anyone has any questions or just wants to talk it over, email me. We can chat. I'm an open book. I'll tell you whatever you want to know about my personal experiences at these studies. And so I hope that this has been beneficial. And if you are on the fence about joining a study, that this has given you encouragement to do it. Join the fight. Do it today. We need you all here in this fight with us. Thank you.
Thank you so much, Bonnie. You are amazing. Thank you for participating in all of those studies. And I will tell you, Bonnie will actually talk with anyone who will help in this fight because she picked me up in 2019. Yes. And you know, my husband also had a variant in TARDBP. And so to have this connection has been incredible.
Yes.
So I will dig into that a little bit more, Bonnie, if we have time. And I'd like to introduce next up my friend Sean McCann, who is living with TARDBP ALS, and he has a very powerful story that he's going to share today. Sean, thank you so much for being here. We are so grateful to have you. I'll let you take it away.
Hello. I just want to thank Nadia for coaching me through this last week. It's my first official Zoom call. I'm not a really big tech guy, so thank you very much, Nadia, for the tutorial on this. Can you guys hear me okay? Okay. So about I'd say November of 2022, I started having fasciculations in the upper portion of my pec. I'm a pretty physically active guy. Ran the New York City Marathon. I cycle a lot, I lift weights. So I figured I had just pulled a muscle. I didn't think anything of it. Then a couple months later, I started jogging, and my foot was dragging, my left foot. So again, I thought it was a pulled muscle or something like that. I didn't think much of it, but my symptoms started worsening as time went on. So by the summertime, I was walking with a little bit of a limp, and I was encouraged to go see a neurologist. So I live in Staten Island, New York, and we do not have a very great reputation for our medical professionals. I'll just leave it at that. I went to one neurologist, and he gave me EMG testing and a few other things. And after about a month and a half of testing, he just shrugged his shoulders and said, "Go see a specialist." I said, "Okay." So I went and saw a specialist on Staten Island. At this point, I had started walking with a bit of a limp, and I started experiencing atrophy in my right hand. I'm a tattoo artist and a personal trainer, so my job started becoming a little bit harder to do. It's harder for me to execute tattoo or do certain exercises. So I had definitely slowed down a bit. I'm like, maybe it's a pinched nerve, and because I had no clear answers, like Bonnie said earlier, when she found out what it was, she had something tangible to hold on to. At this point, I haven't had any of those things, so I'm doing Dr. Google, researching everything, and really just not coming up with any answers. I went and saw a specialist on Staten Island, and he was so sure that I had CIDP, which is chronic inflammatory demyelinating polyneuropathy, which is an autoimmune disease. He's like, "Don't worry, we'll fix you right up." I started going for IVIG treatments, and the first one was miserable. I wound up with blood clots in both of my arms. Had a fever. It was an awful experience. I was in the hospital for 5 days. So he said, "All right, we're going to come back every 3 weeks. You're going to get an infusion. We'll set it up for the home." So I said, "Okay." 3 weeks goes by. I don't hear anything. It was another 3 months before I received my next infusion because they just couldn't, nobody could get on the same page. When I was finally approved, they said, "You have to go back into the hospital for another 5 days because your initial treatment wore off." So I said, "Okay." So at that point, they had given me that plus 60 milligrams of prednisone a day. So it was a pretty awful experience. I'd say from January of 2024 until July of 2024. On June 31st, I had gotten a phone call from my doctor saying he needed to come see me. I need to go see him. I'm sorry. And I went to the office, and he very bluntly just said, "You have ALS." I said, "Oh, what happened to CIDP?" He's like, "Yeah, we were wrong." I said, "Well, how'd you find out?" I had done some genetic testing, but the lab, I guess my insurance company didn't approve the testing, so they withheld the results for 3 months. So I would have had this information in March of 2024, but unfortunately, I didn't get it till June, late June of 2024. At that point, I was frustrated with the medical treatment I was receiving on Staten Island, so I opted to go to Columbia Presbyterian. I had my kidney taken out at Presbyterian, and it was the most wonderful treatment I had ever had. So I was excited to get back up there and deal with people who knew what they were doing. So after I had set up that appointment, I guess in March, and I finally got my appointment in July. So the day after I found out I had ALS, I had actually had my scheduled appointment at Columbia. So I go up there with this newfound information. I think I saw Dr. Shapiro, I believe. And I walk into Dr. Shapiro's office. He's like, "How can I help you today?" I said, "I don't know. I'm dying. What can you do for me?" And he didn't laugh. Nobody laughs when I say that, but humor helps me a little bit. So anyway, he didn't find it funny. And then he said, "Let me see." And I showed him my paperwork. And within 5 minutes, he had made a phone call to Dr. Shneider. He said, "We're going to send you uptown, and we're going to take care of this now." And I was like, "Okay." Now I'm thinking, I have another week until I have another appointment. We'll figure it out. By the time I got down the elevator, Dr. Shneider had called me, and he said, "Can you come uptown right this second?" I was like, "Oh, okay." And at that point, I was with my family, and I was like, "Do you guys mind if we go?" And they were like, "Absolutely not." So they rushed me up to see Dr. Shneider, and I got more answers and more treatment in the hour or so I spent up there with them. And like Bonnie said, it was the first time I had something tangible to hold on to. I knew what I was up against. And I'm like, "Okay, I got it. I understand it now." And they broke it down into such simple, I'm not a really educated guy. So for me to understand what they were saying, it was so helpful, and I didn't feel like I was being condescended to. I didn't feel like anybody was talking over my head. So I actually felt like I had a grasp on what I was dealing with. So they took a skin biopsy. They did a bunch of things. They scheduled me for a lumbar puncture. And like Bonnie said, it's really not that terrible. It was way less awful than I thought it was going to be. And then after my initial appointment, Ellie had reached out to me and we did some genetic counseling. Actually, no, this is my second Zoom meeting. I forgot she did it with me and my mom. And I got even more answers, and I understood what they were trying to do with this, I guess, miracle treatment that they're trying to concoct in a laboratory for me. I asked them if I was going to be like Captain America. They said no. I was kind of disappointed. But she explained how the medicine helps block the mutation and everything that she had touched on earlier. And I'm not a hope guy. I don't live on a hope factor. I've never been that way. I was in the military. My brain doesn't process things like that. I grind it out just one day at a time. But this information gave my mom, my sister, and my dad, and all the people who love and care about me some form of hope, which that's what they need. And that's great. And I'm very happy that they have that. For me, I'm just going to grind it out every single day. And each day I wake up and able to do the things I love. I'm just going to keep on doing that. So I'm excited. My next appointment is on October 4th, and I can't wait to see what else they have for me. I'm sure I'm going to ask Dr. Shneider 1,000,001 questions about alternative treatments and other things. Nadia was nice enough to send me a webinar from, I believe, Duke University also, and it had all these other treatments. So my goal personally right now is to stay healthy long enough in order to receive this treatment and hope that it allows them to make further progress for everyone else. So as long as I keep kicking for the next year or so, hopefully whatever they have in store will be a success, at least on their end. Even if I don't personally benefit from it, if the research can benefit from it and it'll help further the process, it's even better. I have a niece and a nephew, and I hope they don't have the same gene that I do, but God forbid they do. If this allows them some sort of reprieve or kind of ease their mind a little bit, that's, for me, that's the biggest goal. So one other thing I'd like to add, I don't want to take too long. I have a background in physical fitness, so I feel like I have a bit of an advantage for most people who might not know how the body works. So even though I might struggle from things day to day, I know how the muscles and the skeletal system works in different planes of motion. So for me, I'm like, okay, I'm not working in the sagittal plane right now, but I can move laterally in the frontal plane, go up and down the stairs like it's nothing now. And having this knowledge, it allows me to get through my day significantly easier than most people. So if anybody knows anybody who's struggling with ALS in this program and they want a little bit of coaching, please give my information out. I am more than, I know you guys have a trained team of amazing professionals, occupational therapists, physical therapists, and stuff like that. But if anybody needs any questions or any extra help, let me know, and I will gladly help them through the day to day, because I've definitely have a few life hacks on beating this thing. So thank you, Nadia, and thank you, Dr. Shneider and Ellie and everybody else. And I'm looking forward to hearing the rest of the webinar. Thank you.
And happy birthday to your to your mother, Sean.
Ellie, I apologize for you having to deal with my mother. I know she's a bit much.
I'm glad that you were able to join us today, even though it is her birthday. So
Yeah.
thank you.
Thank you very much.
And thank you for your amazing attitude. I think, we all help each other through this disease. So it would be wonderful that you're open to talking with people. All right, and we may come back to you for questions in a bit, Sean.
Sure.
And next up, I'm going to introduce Dr. Neil Shneider. Dr. Neil Shneider is a physician scientist with expertise in neuromuscular development and disease. He's a graduate of Harvard College and of the MD PhD program of the Columbia University College of Physicians and Surgeons, and he was chief president of the Harvard Longwood Neurology Training Program. Dr. Shneider completed his graduate work in the lab of Dr. Richard Axel and his postdoc in the lab of Dr. Thomas Jessel. After several years in the intramural program of the NINDS, Dr. Shneider returned to Columbia University as assistant professor of neurology and in the neuromuscular division. He is a member of the Center for Motor Neuron Biology and Disease and of the H. Houston Merritt Clinical Research Center. Dr. Shneider has clinical expertise in the diagnosis and care of patients with ALS and related motor neuron disorders. It is a real pleasure to have you. Thank you so much, Dr. Shneider. I'm going to let you go ahead and screen share and take things away.
Thank you, Nadia. Hope you can hear me. And thanks to Ellie for that great introduction, and to Bonnie and Sean for their inspiring comments. It's what makes what we do so meaningful. And I look forward to seeing you on the 4th, Sean. Bring your mom. I'm going to share here. I'm trying to anyway. Can you see that?
We can, yes, perfect.
Wonderful. Well, I'm going to pick up a little bit from Ellie and talk to you specifically about TARDBP and some of our efforts at Columbia and through Silence ALS to address the specific concerns of individuals and families with ALS causing mutations in the TARDBP gene. As Ellie told you, TARDBP, TDP43 is one of dozens, 30 or more genes that have been implicated in the genesis of ALS. But TDP43 has a very special role in ALS, more so I would dare say than any of the other ALS genes, because of what we believe a very clear central role it plays in this disorder. We first heard about TDP43 in 2006 when Virginia Lee and John Trojanowski at the University of Pennsylvania and their colleagues and other labs here highlighted working in Japan identified TDP43 as a major component of the abnormal aggregates or inclusions, neuronal and cytoplasm and glial inclusions that we find in degenerating neurons in ALS. And some of these are shown here. I hope you can see my cursor. But in neurons, but also non-neuronal cells in the nervous system, in ALS, but also in frontotemporal dementia, which you heard is a disorder that's clinically and in many ways related to ALS. We see these abnormal inclusions shown here, and they come in different forms. Here you can see diffuse staining of TDP43 in the cytoplasm, whereas normally TDP43 is located here in the nucleus, which I'm circling. TDP43 can be mislocalized into the cytoplasm, and it can form these aggregates. You can see these sort of dark staining collections of brown staining here in the cytoplasm. And in some cases, we see these very dense, rounded inclusions or aggregates in the cytoplasm, which are highly abnormal and a hallmark of ALS and frontotemporal dementia. These are seen not only in the spinal cord, but also in the motor part of the brain and the motor cortex. You can see here an abnormal TDP43 positive inclusion in the cytoplasm of this degenerating upper motor neuron. So this was described in 2006, and people began looking for them. And what we learned pretty quickly was that TDP43 pathology was not seen in a small fraction of ALS, but actually in a very large majority of ALS cases. And when we looked at cases, post-mortem cases that we had in our laboratories around the world, it was demonstrated that TDP43 pathology is present in over 95% of ALS cases and about 45% of frontotemporal dementia, FTD, cases. So this represented a significant finding in the ALS and FTD communities, but it wasn't certain at first anyway whether this was just a marker of pathology or whether TDP43 played a active role in the onset and progression of ALS and FTD. But that any doubts about that, I think, went away in 2008 with the discovery that mutations in TARDBP, encode for TDP43, the mutations in this gene were genetically associated with ALS and frontal temporal dementia. So this wasn't just a pathological finding and a marker of disease progression, but mutations, so-called pathogenic mutations in this gene could cause ALS and frontal temporal dementia. This clearly, squarely implicated TARDBP, TDP43 in the genesis, the pathogenesis of ALS. Now, these mutations were extraordinarily rare. You heard from Ellie, she said 5%. I would say it's even a smaller percentage of all ALS patients, 2 to 3% max, who have a TARDBP mutation. So the other, to me, significant finding of this was, even though these mutations were very rare, ultra rare, the majority of ALS patients, ALS and FTD patients, had TDP43 pathology, which meant that the genetic cases and the biology of those genetic cases were teaching us something about the biology of ALS more generally. And I would say that the study, therefore, of these rare genetic forms of ALS, I think this is true for TDP43, but also for the other ALS genes, but these rare genetic cases can teach us something about the broader family of ALS. So the question really then was, how is it that these mutations in TARDBP cause ALS? And secondary question is how is TDP43 more generally involved in the pathogenesis of ALS? So I would begin by telling you, and as Ellie began to say, a little bit about the function of TDP43. TDP43 is a DNA and RNA binding protein as part of a very large family of these DNA RNA binding proteins. It functions in a variety of ways in RNA metabolism. I told you it's predominantly present here in the nucleus, but it's also found in the cytoplasm to a much lesser degree. In the nucleus, it plays an important role in the synthesis of RNA from DNA, the so-called transcription of RNA. It's involved in the processing of those RNAs, so-called splicing of RNAs. It's important for RNA stability, transport out of the nucleus, and it helps to control within the cytoplasm the translation of those RNAs into protein, both in the cytoplasm and even in more distal regions of the neuron and the axons close to where they contact with muscle or other neuronal partners. So TDP43 plays a lot of very critical function in neuronal, I think, development, function, and longevity. And of course, the question then was, do these mutations in some way result in the onset of ALS, or is there another explanation? I would tell you that we don't know exactly what the mechanism is by which TDP43 and the mutations in the TARDBP gene cause ALS, but we can really, I think, summarize it in one of two ways. Either these mutations lead directly or indirectly to a loss of function in TDP43, and that loss of function leads in ways that we don't fully understand to neurodegeneration. And there is very good evidence for TDP-43 loss of function. I told you that TDP-43 mislocalizes in neurons, again shown here. And in the process of mislocalization, TDP-43 is excluded from the nucleus. So if TDP-43 is not in the nucleus, it can't do what it normally does in the nucleus, which is, again, to control RNA metabolism. And there is very good evidence that's been evolving even very recently, in recent years, demonstrating that clear evidence for TDP-43 misfunction, dysfunction in the nucleus. And some of the targets of TDP-43 activity are abnormally regulated in the course of ALS. Those are at the very least markers of pathology in ALS and frontal temporal dementia, and in all cases where this TDP-43 pathology. And it remains an open question the degree to which that misfunction or dysfunction contributes to neurodegeneration. But it's a very active area of research. So loss of function is one possibility, but another possibility is that these abnormal localization of TDP-43, these large inclusions and the mislocalization into the cytoplasm causes a gain of function, a toxicity that's not necessarily associated with the normal function of TDP-43. So the gain of function versus loss of function, these are not exclusive. Both could be true, and we have to take these both of these possibilities into account. So here's just a little schematic that I pulled from a paper that I like. TDP-43 here in red is normally a nuclear protein, both in neurons and in non-neuronal cells. In the course of disease, TDP-43 mislocalizes into the cytoplasm, and there it is abnormally processed. Ubiquitin is a marker of the abnormal processing of TDP-43, and this ultimately leads to the formation of these abnormal aggregates or inclusions in the cytoplasm, which consolidate over time into these more mature inclusions shown here in yellow, and either the loss of function from the nucleus and or the presence of these aggregates we believe is toxic to neurons. So the question really then is, you know, how does it come about? How is it that TDP-43 with a mutation, and we believe even without mutation, results in the mislocalization of this protein and the abnormal aggregation? So there's a lot of science in here, too much for a webinar of this kind, and I'm happy to answer questions of this, but I try to simplify this process in this diagram. Normally, TDP-43 and other DNA-RNA binding proteins exist and function in solution as single proteins floating and acting within the nucleus and possibly the cytoplasm. But TDP-43, because of particular structural features, has the ability to what we call phase separate, meaning it can form liquid droplets, sort of like oil in water. And these liquid droplets exist and are part of the normal functioning of TDP-43 and related proteins. And within these droplets, there's a very high concentration, we believe, of these of TDP-43. And what we believe is true, that mutations in this gene that are associated with ALS FTD cause an abnormal misfolding, an abnormal condensation or aggregation of these proteins within these droplets. And this misfolded protein can go on to form not liquid-like droplets, but more gel-like droplets that can further consolidate into these insoluble aggregates, these insoluble fibers. And somewhere along the way here, some species is formed that is toxic to neurons and leads to the beginnings of degeneration of neurons in the brain and in the spinal cord. So there are a lot of efforts in the field, and I imagine there'll be lots of questions about it, but there's many ways to think about perturbing this pathway. And some of them are written here. You can think of normal drugs, small molecules that might inhibit the formation or speed the dissolution of some of these abnormal structures. You could play with RNA that clearly plays a role in the formation of these abnormal condensates. You can think about enzymes, using enzymes and other molecules to actually actively break apart these aggregates and return the protein to its normal soluble monomeric form. And all of these things are being pursued in academic laboratories and in industry labs because this is a very promising therapeutic target. This is, we believe, the most early steps, the most, the first steps in a process that leads ultimately to the degeneration of neurons. And the idea is if we could stop this, we could slow progression, halt progression, and in the cases of those who are maybe not yet asymptomatic, maybe even prevent onset. So we at Columbia and through the efforts of Silence ALS have taken an approach to this problem. And this really is based on the idea that the abnormal misfolding and the formation of these aggregates depends on high concentrations of these proteins. So the idea that we had was to lower, if you can see here, if we could lower the amount of TDP-43 in the cell, this could lower the concentration of TDP-43 within the condensates and prevent the misfolding that is occurring and which leads to the formation of these abnormal toxic aggregates. And the idea is that with that, we could slow the formation of these aggregates and allow cellular machineries that are normally and naturally present to eliminate these aggregates and restore the system to its original state, slowing progression, ideally halting progression and allowing for some degree of functional recovery. That is, in theory, what we are trying to do. So how do you lower levels of TDP-43? Ellie introduced this. There is technology available that's already been applied to ALS to lower specific protein levels using ASOs, antisense oligonucleotides. Ellie mentioned this is the class of drug that is used in SOD1. This is the first gene-based drug for ALS, tofersen, now called Qalsody. This is an antisense oligonucleotide designed by Ionis Pharmaceuticals and developed by Biogen and approved last year for people with SOD1 ALS. Ellie mentioned we're pursuing something similar for FUS ALS for people with mutations in fusion sarcoma. And we thought that we could apply a similar technology to TARDBP. And here, an ASO, you can see shown here, so-called GAPMER. This is a DNA RNA hybrid, which is specific for a specific sequence within the TARDBP gene in this case. And what these the specific class of ASOs does is it activates an enzyme, a cutting enzyme called RNase H, and RNase H then destroys the target to which the ASO is bound. You destroy the messenger RNA and you destroy thereby or lower the levels of the protein that is the product of that RNA. That's the simple idea of antisense oligonucleotides. So in the case of SOD1 and in the case of FUS, these were so-called non-selective ASOs, meaning that they didn't distinguish between the wild type and mutated form. So this is a schematic similar to what Ellie showed you. She told you that we inherit 2 copies of every gene, including TARDBP. We have many people on this call, and many of the TARDBP families that we work with have 2 copies. One a wild type or normal copy, and another a mutated copy that bears a pathogenic mutation in TARDBP. The DNA is made into RNA, and the RNA is then translated into protein, both the wild type form here in these blue squares and the mutant form, which I've depicted here in red triangles. And in the course of disease, as I've told you, these proteins aggregate in an abnormal way to form structures that are toxic to neurons. So what, again, they did in the case of SOD1 and what we're trying in case of FUS is develop a non-allele selective ASO, that is an ASO that doesn't discriminate between the wild type and the mutant form. And the consequence of that would be to lower both the levels of wild type and mutant protein. That's the product of that gene in case of SOD1 and FUS. And the idea of that is by, again, similar to what I described to you, by lowering the levels of the protein, we slow the formation of new aggregates and allow for the dissolution of existing aggregates in a way that would be clinically beneficial. The problem with TDP-43, however, was that TDP-43, as I told you, is a critical protein, right? It does many critical things, and the concern was that if we lowered TDP-43 too much, that we could create more problems. We could actually exacerbate a patient's neurological situation by creating new problems through an induced loss of function, right? So what we did to get around that problem, as I'll tell you, is to design an antisense drug that only or selectively recognizes the mutated copy of TDP-43, largely leaving alone the wild type copy. And the consequence of this was that we would selectively lower mutant TDP-43, leaving normal levels of TDP-43 present to do its normal function. And the hope is that that would still, result in a dissolution or elimination of some of these aggregates and subsequent clinical benefit. So that was our strategy. And to do this, we partnered with the Eleanor Foundation. This is a nonprofit organization that was founded by Stanley Crook, who was a founder of Ionis Pharmaceuticals, and in his retirement, he decided to bring this technology to bear on ultra-rare, he likes to say nano-rare cases of genetic disease. That is forms of genetic disease that are really too rare to attract the attention of a drug manufacturer, pharmaceutical or biotech company. And he understood that this really could only be done on a nonprofit basis. So we partnered with him and with the Eleanor Foundation that he established to found this initiative called Silence ALS, which was aiming really to discover and develop personalized investigational ASOs for ALS patients with rare pathogenic mutations. And really, this was an effort to build on Eleanor's platform and to apply the technologies and the infrastructure that they had developed and apply that to these ultra-rare genetic cases of ALS. And in this, we were really leveraging our own expertise and experience in ALS and applying Eleanor's approach to ALS families in a very focused way. And really the goal here was to serve the underserved, to serve a group of ALS patients who really, I think, couldn't expect to attract the attention of drug makers. And of course, our main goal was to help these individuals and families. But as I said earlier, our larger goal was to understand through the study of these TARDBP and other ultra-rare genetic ALS cases, understand something more broadly about ALS and to apply those learnings to the larger ALS population. And we have begun on this journey with and Laura, that's been really an extraordinary partner in this. We got FDA approval for our first ASO in 2022, and this was for a TARDBP mutation carrier. This was a 40-year-old woman, mother of 2 young children, who had no family history but had a de novo or spontaneous mutation in TARDBP. And we worked with Eleanor to develop an antisense drug called TARD-001, Eleanor TARD-001. This was an antisense oligonucleotide that had, you can see here, 55 times selectivity for her mutated copy compared to the wild type. So this was, as I told you, a non-allele specific antisense drug. And I won't go into any details except to say that Eleanor did an extraordinary job and developed this drug in about a year. From the beginning of the process, we were able to treat our patient with 4 or 5 doses of this ASO. It was very well tolerated, but unfortunately she had declined significantly in the year during which this drug was being developed. She had a very aggressive form of the disease, as we sometimes see with TARDBP cases, and she had an event that made it impossible for her to travel and continue in the program. So her treatment has stopped, but we learned a great deal from her, and we've applied learnings from this case now to a total so far of 18 cases of TARDBP ALS that have been, including hers, that have been reviewed and accepted by Eleanor for ASO development. These programs, some of them are now withdrawn for various reasons and not always successful, but 15 of these programs are active and at various stages of development, and we're hoping to treat our second TARDBP case later this year, applying now for FDA approval. As Ellie said, when we meet individuals who are symptomatic with TARDBP mutations, as is true for all ALS genes, this is an entree into a family. We use this to identify individuals who are carriers of these mutations and are at risk. So far, we have identified over 25 asymptomatic carriers of TARDBP mutations in the US and around the world, including some, as is shown here in green, from one very large TARDBP family. Our goal here, in silence initially anyway, is to treat symptomatic individuals with an individualized ASO. But our goal is in following asymptomatic people is to identify them and as early as possible in their disease course, ideally before symptom onset, but after the disease has started to manifest. That's one of the goals of the ALS Families Project. And we are beginning to study these families through the Pre-Silence ALS program to begin the early steps of ASO design and development, so that if and when an individual converts, that we have a leg up. We've saved a lot of time so that we can bring an ASO to the clinic much quicker, earlier in their course, when they're most likely to benefit from treatment. So part of our goal here today through this webinar, is to communicate with TARDBP families. Some of you are here online. I'm sure some family will see this webinar later, and I encourage you to reach out to Ellie to inquire whether or not your case would be appropriate for inclusion in the ALS Families Project, in the pre-silence ALS program, and if there are any symptomatic individuals with TARDBP, we encourage you to reach out as well. Our hope is that as we build a library of these TARDBP specific ASOs, that we will be able to get therapeutic ASOs to individuals much faster. We have a lot of work to do ahead to demonstrate that these ASOs are first safe and effective, but we have a lot of hope for this program and have funding now from the Urgent Network of the NINDS from NIH, and are seeking additional funds to expand our efforts and include more patients with TARDBP and other ultra-rare ALS mutations in the Silence program. So I'll stop there and take some questions online, and thank you very much to End the Legacy for organizing this event and including us in this important work. Thank you.
Thank you so much, Dr. Shneider. That was incredible. I also wanted to just share that we had another family, Isabel and Gilles Gayo, who are being seen at Columbia, who really wanted to be here today, but we're traveling and they're across the world right now. But we had a wonderful discussion just right before this webinar started, where they were sending their support, and we were all chatting about how far we've come in the TARDBP ALS world in finding each other and making these treatments happen. We have several questions. What is the status, Dr. Shneider, of TDP-43 biomarkers getting to clinic? And would those be the same for sporadic ALS and genetic versions of ALS?
Right. So over the last a couple of years, there has been a great deal of work, exciting work, from a number of groups that have identified some of the targets of TARDBP, of TDP-43 in the nucleus. One of the things that I mentioned that TDP-43 does is it is a regulator of splicing, of the processing maturation of pre-mRNAs to mRNAs. And when that function is perturbed, when TDP-43 is not in the nucleus to do what it's normally supposed to do, those regulated events go awry. So in some cases, proteins that are inhibited or suppressed by TDP-43 go up. And in other cases, proteins that depend on TDP-43 for their expression go down. So a lot of work has gone into identifying these TDP-43 regulated proteins, and they are being used now as potential biomarkers for ALS. These are not relevant just for individuals who have TARDBP mutations, because I told you 97% of ALS cases, 45% of FTD cases have TDP-43 pathology that involves nuclear exclusion and TDP-43 loss of function. So in the vast majority of ALS cases, these markers of TDP-43 function are potential markers of disease. And as I said, these are very early events. So there's a real potential of using some of these markers as pre-symptomatic markers of disease onset. And there are many groups, both in industry and in academia, that are working to exploit these findings and to develop assays for that could be used as symptomatic, diagnostic markers, but also as a way of identifying response to therapy, right? Because the idea is if you could restore TDP-43 function in the nucleus, these markers should go away. So they're not only good for diagnostic purposes, they may also be good as a marker for response to therapy. Very important work.
Thank you so much for that. And also for people who might carry the gene, what is the kind of the plan for silence ALS? Will pre-symptomatic carriers be treated at some point in the future? Are we studying in people with ALS right now? How will the development of that work?
Right. So, I mean, in theory, if you had an ASO or any drug that was safe and effective in treating the TARDBP ALS, in theory, that drug may be useful to prevent the onset. Right. When you have an ALS-associated mutation in TARDBP or FOS or SOD1 or any of these genes, people don't get ALS when they're 2 or 3 or 10 or 20, right? It takes years to develop. So these mutations increase the risk of developing ALS, but you don't again need a mutation to get ALS. The large majority, 85+% of people don't have a mutation. It happens with wild type protein as well. But the mutations increase one's risk. If you could eliminate the mutated gene or the expression of the mutated gene, in theory, you could normalize somebody's risk of developing ALS. So that, I think, is the goal. But we have to get there in steps. I think we first have to demonstrate to ourselves, to the FDA, to our patients and families that these drugs are safe and effective. And then we need to walk this back, from symptomatic individuals to pre-symptomatic individuals who are showing early signs of disease by biomarker or EMG or neurological examination. And I think then with confidence that these drugs are safe and effective, we can begin to think about pre-symptomatic treatment. And that is, I think, the stages of development that are necessary to move this forward.
Thank you. I know that's a tough question. So thank you so much for taking that on. And Ellie, could you talk a little bit more about what asymptomatic family members should expect through the study that you have?
Sure. So baseline research participation can happen for anyone who's at risk, so has a first-degree family member with a known ALS-causing mutation, or a strong family history of ALS, 2 or more people with ALS, including one first-degree family member. And kind of baseline participation is coming into a prevent site. So there are 34 sites around the US. I think someone had a question about Texas sites in the chat. There'll be 2 Texas sites opening up. And they're usually half-day visits. That will include blood collection, urine collection, vital signs. We'll do some small cognitive evaluations and testing, breathing examinations, a full neurological exam. We'll do strength testing on specific machines. And then there'll be an optional skin biopsy, optional lumbar puncture, and optional electromyography and nerve conduction study. The EMG very much depends on what site you're attending. Some sites don't have the ability to conduct EMGs at their specific locations. So those appointments happen on an annual basis. We're also collecting blood sampling done remotely. I'm forgetting the frequency of that, but I think it's at a 6-month interval that will be set up. You don't have to come into the local center to do that. And then at any point throughout your participation, if you haven't undergone predictive genetic testing, you can always opt into the genetic testing arm. So that you would let your site know that you're interested in talking with a genetic counselor. Then you'd be referred to our kind of genetic counseling team. We'd have an introduction call, talk about kind of the basics of the process of genetic testing, and then everything is done remotely. We'll have a video appointment, really reviewing all those considerations I mentioned, talking about your motivations for testing, benefits and drawbacks. We'll send a saliva collection kit to your house to provide a DNA sample for the genetic testing, and then meet again over video to disclose your genetic test results back to you. If you test positive, of course, you'd continue to be part of the PREVENT study. And if you have test negative, you can continue on as a control participant as well.
And then, Ellie, do people need to find out the results of their genetic testing, or can they continue to participate in the study even if they don't know the results of the genetic testing?
Good question. Yeah, so you can continue in your annual research participation without ever going through genetic testing. We call that non-disclosure participation. So certainly we have many individuals who aren't ready or are not wishing at this time to learn their results, and we welcome those individuals as well.
Perfect. We're getting some questions about locations and contact info for some of these studies. You will be able to find those on the End the Legacy website, endthelegacy.org as well, just so that people know that. Dr. Shneider, we have a great question in chat that was kind of referencing that since these ASOs are specific to variants, how do we think that regulatory approvals might move forward for this? I don't know if you can speak to any of the, maybe, how might this need to be handled differently than a treatment that might, benefit broadly as it moves ahead?
Right. So in 2021, the FDA issued a series of guidances that really made the silence ALS, the NLORM approach possible. We began, my entree into the ASO world was with FOSS before these guidances were issued, and we had to break a lot of ground to get an N of 1 ASO study approved. Because of that effort and others, including that of Tim Yu and many other early efforts in this area, the FDA issued these guidances that are specific for the ASO technology for this particular chemistry. So it doesn't apply to say other gene therapies or other chemistries, and it's specific for these ultra rare genetic forms of the disease. So the guidances were issued, and in our process, we follow those guidances very carefully, and we have had expected success. The FDA approved the NL-TARDBP 001 ASO, and we have a second ASO now from Silence ALS for patients with CHCHD10 mutations. In that case, actually, because of the specifics of that gene, we did not have to make an allele-specific ASO. It's non-allele specific, and we are preparing to treat 10 patients total so far with that particular ASO, and we expect similar success with the other TARDBP and other Silence ALS ASOs when they are IND ready.
Thank you very much for that. And then, are there ways for, not everyone may choose to be followed in a research study, or they may not know about research studies. So are there ways that And maybe as we're getting the word out and the legacy and creating more awareness that there are genetic mutations in ALS and more people are getting tested. Are there ways for people to be followed outside of a research setting? And might that be a useful thing as we move forward? I think that question's both for Dr. Shneider and Ellie.
You want to go, Ellie?
You can go ahead. It's a tough question.
It's a tough question. You know, I think that these are research INDs. This is cutting edge technology. I'm speaking now about the Silence ALS and the individualized ASO programs. These are research efforts that are centered in major academic centers like ours. Through All ALS and Prevent ALS, we have a new consortium here in New York State, the Empire State ALS Alliance. Through efforts of this kind, we're trying to reach out to non-academic centers and to expand our efforts into communities where people don't have the same kind of access, and enable our colleagues in non-traditional research centers to participate and refer patients to these efforts. We're trying to broaden these efforts, and I think we're succeeding. You know, Silence ALS is a small program. It's an extraordinarily expensive effort, and so we're focusing on, by necessity, on small numbers of people. But our hope is, as I said, as we develop a library of these ASOs, we can reuse them, repurpose them. Even though they're individualized in their development, they will potentially help other TARDBP patients, and we can expand and broaden our efforts at that point. But yes, I mean, there are organizations, ALS TDI, Synaptic Cure, other groups that are not, research center based, that are contributing to our research ecosystem, and I'm hopeful that more and more patients and individuals at risk can participate.
Thank you so much. Sean, Bonnie, do you have any last thoughts before we close things out here?
I have one. We do have a TARDBP Facebook group online. So if this affects your family, please join us. We are one big family. We are all in this together. We share as much information as we possibly can. And we just try to hold each other up every single day. So that's something, please look for that online.
Sean?
Just trying to figure out how to unmute. No. Oh, actually, I do have one question. I think you said the location of my mutation was in 382T, I believe. Is that correct, Dr. Shneider?
As much as I remember you, Sean,
It's fine.
the
reason
I don't remember your specific mutation, but
though I was looking at your charts before, and you had the 18 people who have it, and each one had a different one, and there was somebody else with the 382T. I guess their mutation was at the same location as mine.
Important point that these ASOs don't necessarily target the mutated nucleotide, the mutated position. There can be a change on the RNA that is linked to, but not exactly at the mutation, and that can be used. So you can have 2 people, 5 people with the same A382T mutation. Each would require a different individualized ASO, and the same is true that an ASO that is used to treat an somebody with your mutation may be able to treat somebody with a entirely different TARDBP mutation. And that's part of what we're learning in the process as we study more and more patients, both symptomatic and asymptomatic. We're identifying more of these potential target sites that we can direct an ASO towards that could be used repeatedly for families and individuals.
Okay, thank you.
Thank you. And with that, we are at the end of the hour. It was a wonderful webinar. I am so grateful to our presenters. If you would like any more information, please feel free to reach out to us at info@endthelegacy.org. A very warm thank you to everybody who attended and presented today. You will also be receiving a post-event survey. And so please take the time to let us know what you thought about today. And I hope that everybody has a wonderful evening. Thanks so much for attending all.
Thank you. Take care.